Hello, welcome back. In this lecture, we're going to look at a polymerase chain reaction or PCR. I know when we started learning about biochemistry, I promised you we must learn about PCR and its importance in medicine. And PCR has come to the forefront. I know for us in medicine, we knew it for HIV, paternity testing, but now it's come to the forefront a lot because of COVID-19. So PCR is applicable under this series of gene expression and regulation because it is based on the principles of DNA replication. Okay, we remember in DNA replication, you had the double-stranded being opened by the helicase enzyme, primer binding, and then polymerization by the DNA polymerase enzyme, and the whole DNA was copied to create two daughter strands, one containing the parent strand and one the daughter strands. So this is what we're going to see happening in polymerase chain reaction. So what is PCR? So this PCR is a technique that has been used to amplify DNA segments. So amplify is to increase the copy. Where it's the essence of amplifying. Sometimes, like for example, a good example when there was HIV, you could not be able, if someone was infected with the virus, and the predominant techniques depended on seeing the antibodies which are produced by the body after the virus has been there for a long period of time. However, in the early phases of infection, we might not be able to see the antibodies being produced because they take some time to be produced, as long as 6 weeks. So even if there's one copy of the virus, of a virus or HIV virus, PCR can be able to amplify this segment or this copy of DNA and allow it to be identified as being present. So the PCR chain reaction, it came about, it was allowed amplification of specific DNA sequences from very tiny samples. And the technology was invented by Carrie Mullis in 1985. And as we know, it has been widely used in diagnostics, in genetic research, in forensic medicine, and also in infectious disease like COVID-19 detection. So we will find that PCR really requires amplification of DNA at high temperature, and at the beginning, the temperature, maintaining this high temperature was a challenge. However, with the invention of the machine, the thermocyclers, the automated thermocyclers allowed the PCR technology to occur much easier. So let's look at the components of the PCR. What is required for us to amplify DNA in the thermocycler? So of course we require the DNA template. So where we have our target DNA sequence that has to be amplified. So the DNA template could be maybe that a sample that we suspect having HIV, or sample we suspect that maybe having COVID-19, or any other sample that we suspect that has the DNA that we want to amplify. The other thing is primers. So we do know polymerase enzymes do not start synthesizing DNA, they only add nucleotides to an existing DNA sequence. So that's when we require primer. And these primers usually are very specific for the gene of interest we want to amplify. For example, if we want to detect the presence of COVID-19, we have to have primers that are very specific for COVID-19, such that when the DNA opens up, the primers are able to bind to sequences that are present only in the the DNA particle or the gene segment specific for COVID-19. So that's why we're saying they are short single-stranded sequences that flank the target region. Then we have our DNA polymerase. In this case, we use a DNA polymerase called Taq polymerase that is heat stable, and is able to work under high temperatures. It was isolated from some bacteria that lives in the hot springs. So it is able to polymerize DNA at high temperatures. And of course, we require the nucleotides. In this case, they will be deoxynucleotrophosphates like deoxy-ATP, deoxy-GTP, deoxy-TTP, and deoxy-CTP. So this will be the ones that will be used by the polymerase enzyme, will be added on the 3' hydroxyl end during elongation. Then we of course require a buffer solution to provide the adequate condition for the Taq polymerase, and magnesium ions, which are usually cofactors required by the DNA polymerase, and of course our thermocycler. So all these things are mixed in a PCR tube and they are placed in the thermocycler. So what happens? Let's look at the steps in the PCR, in a basic PCR. So the first thing is, once the tube has been placed, the first thing is denaturation of the DNA. So we do know at high temperatures, the double-stranded DNA gets denatured, where the hydrogen bonds that were holding the nucleotide on the respective strands are broken. Okay, and so we end up with 2 single strands. So the essence of denaturation is to allow these nucleotide sequences that are present to be accessible to the primers, such they can be able to be elongated. The second phase after denaturation at 94 to 95 degrees is annealing. So here we bring the temperature down to around 50 to 65 degrees. This is a range because the annealing will require on the type of primers that are being used, or the melting temperature of the primers. We're not going to use to reduce the melting temperature or the reannealing temperature of the DNA, but of the primers. Okay, but we don't want the DNA to reanneal. All we want is that the primers should attach. So that's why the temperature is around 50 to 65 degrees. So here, of course, because the primer design has been created in such a way, it will definitely bind to the DNA if it is present. So when we bring the temperature to around 50 to 65 degrees, we do know for sure the primers will anneal to the correct sequences on the target DNA. The next step now is elongation. We bring the temperature back to up again to around 72 degrees. And this is the optimal temperature for the Taq polymerase. So after the primers have already bound, the Taq polymerase will synthesize the new DNA by adding new nucleotides at the 3' hydroxyl end. We've learned about this, how they do that. So just like the DNA polymerase we learned about, similarly the Taq polymerase will operate in the same way. So this will result in, now if we started with one strand, you can see now we'll have 2 double-stranded DNA that was resulting from one original double strand. Okay, so the fourth step is cycling. So this process is repeated. So you can see in one cycle we are ending with 2 copies. In the second cycle, each of these will end up with 4 copies. So imagine these cycles are repeated so many times, around 25 to 35 cycles, and this means that even if there was only one copy of a particular DNA segment we wanted, we have it amplified so many times, and we can have millions to billions of copies of the target DNA, which can just be produced from one molecule. So this is the essence of the PCR. We are able to amplify the copies of DNA to allow not only quantification, but also detection of whether that particular DNA is present. Let's look at the types of PCR. So what we have described is the conventional PCR. So this is the most basic form of PCR, and all that is done is just to amplify the sequence of DNA. Once the DNA sequence has been amplified, how do we detect it's there? Here we can use gel electrophoresis. Here is an example that I got from a site in ResearchGate. Here is its site. So in this, we can see we had several samples labeled 1 to 8. And here in the middle is what we refer to as the ladder or the standard DNA. So once the DNA is amplified and put in the gel, the fragments will move according, and here we can be able to identify the presence of particular DNA corresponding to the gel. Now the conventional PCR cannot tell us how much DNA is present. It only indicates that the particular fragment of DNA you are looking for or you hoped to identify is present or or absent. For example, here you can see in the sample 1, we're not seeing any ladder corresponding to anything on the gel. We could say this sample is negative. However, we look at sample 2, we have some of it, 3, 4, all of it have something. But if you look at 4, might not be corresponding to a particular part on the gel, although we have some DNA. If you look at 6, 7, 8, probably this is the similar to either the upper one, either this big one, maybe the concentration is low, but we cannot tell from the gel. So the conventional PCR only tells us this particular DNA segment is present in the sample, but it does not quantify. So this one is used in cloning, in gene identification, or in DNA fingerprinting. The other type of PCR is what we refer to as real-time PCR or qPCR. So this is different from conventional PCR because it quantifies the amount of DNA that is present. So it allows real-time monitoring of the DNA amplification, and as you amplify, we're able to tell how much DNA is present. So how does it do this? It uses fluorescent dyes such as SYBR green that are able to bind to the double-stranded DNA. So for example, at the beginning, if we have only one copy of the double-stranded DNA, the fluorescent dyes will bind to it and will produce fluorescence. Okay, so in the sample, we are going to add some of this fluorescent dye, and the fluorescence that it will be produced will be dependent on how much of the target DNA is present. As we do the PCR and we amplify the DNA, okay, the amount of amplified, the intensity of fluorescence will correlate to the amount of the amplified DNA. For example, here we had one copy of DNA, then it is amplified, and then now we have 2 copies, and we can see we have more fluorescence being produced. So the good thing about this, we can measure the curve and we can see as the amplification is continuing, the numbers of copies of fluorescence is increasing because more fluorescent dyes are binding to the double-stranded DNA. So the our real-time PCR provides quantitative data on the DNA concentration in real time without need of a post-PCR gel electrophoresis. So this one was very important in the detecting of COVID-19, even detecting low-level pathogens or measuring gene expression. The third type of PCR is a reverse transcriptase PCR. So we do know some viruses, their genetic material is RNA, for example, the HIV virus or the COVID-19 virus. So when you're amplifying, you might be looking for DNA to amplify, but in that particular sample, you might not find a particular DNA for this organism. For example, in HIV, we know the genetic material is an RNA. And so when we want to amplify this RNA, what do? We first convert the RNA to a cDNA or a complementary DNA. It's called complementary because it's complementary to the RNA. So this is done by an enzyme referred to as reverse transcriptase, okay, which is able to synthesize the cDNA from the RNA template. So what the reverse transcriptase does is that it uses the RNA template to make a DNA strand. Of course, it requires the primer, then it adds the deoxynucleotides to the primer, complementary to the base pairing, of course, in accordance with the Watson and Crick base pairing rules. So it creates a complementary cDNA. So the complementary cDNA then is amplified through traditional PCR. So now we are able to say, oh, we can find a DNA that is very similar to the RNA of the virus. And this is what we are saying it is used for detecting RNA virus such as HIV or even SARS-CoV-2. So we use this RT-PCR not only in detection or diagnostics, but also in gene expression, studying gene expression profiles. Because, for example, we are seeing that maybe you're seeing a messenger RNA and you want to see which particular gene coded for that messenger RNA so we can make the cDNA. And then from that cDNA, we are able to go back and look where do we find this particular cDNA. It's on which chromosome, which segment of the chromosome. So that's why it's called to studying gene expression profiles. Then we have the multiplex PCR. This is a very interesting one because sometimes you want to find multiple pathogens in a sample, or you suspect multiple infections. We want to check multiple genes, for example, in diseases where multiple mutations can contribute to the pathogenesis of the disease. So a multiplex PCR amplifies multiple DNA targets simultaneously in just one PCR reaction. So what makes this possible is because we add more than one set of primers. So in that particular sample, we add more than one set of primers, and each different primer will amplify the target sequence within the same reaction tube. So the advantage of a multiplex PCR is that it can save time and resources by allowing detection of several pathogens or genes at the same time. So we can see here we can have pathogens, different pathogen A, B, and C, and we want to check the particular sample if these particular pathogens are there, maybe a co-infection. So what we do, we have primer sets that are specific for gene sequences in each of the pathogen. And when the primers are added and we amplify the DNA that is present through gene electrophoresis, we can be able to identify if we have segments or we have amplified DNA of pathogen A, B, and C present in the sample. So multiplex PCR is used widely, as we said, in genetic screening, for the infectious diseases. For example, we want to check multiple pathogens in a sample, or in forensic testing, for example. You have a sample and maybe you've collected a sample let me give a bad example. Maybe it's a rape and you've collected some specimen from the victim, some semen, and you're not so sure who is there, who is the offender, but you have several suspects, and you can check if this semen belongs to several people, and you can amplify the using primers from different people. You can be able to say if this sample contains DNA from X, Y, Z. This is not the best example, but yeah, this is an example that you can use for forensic testing, or even to know people who are in a crime scene. Okay, you might have something from the crime scene and you want to see how many people were there. During forensic testing, you can be able to amplify several and find out how many people were several suspects were there at the same time. So that is it for our lecture on PCR, and you can see the PCR has multiple applications, from detection of genetic disorders such as sickle cell to cloning of complementary DNA or nucleotide sequencing, to detection of mutations and modifications in DNA, identifying a disease genetic tendency, imaging and diagnosis, and even treatment of viral infection. So we're able to check if the patient has recovered from COVID by doing another PCR, checking presence of pathogens, recognizing emerging infections or infectious diseases, microbes detections, forensic, and finally cancer. So it has multiple applications. Thank you.